You are all most welcome to this seminar on pesticides and pollinators. This is jointly organized by FRAM, the Center for Future Chemical Risk Assessment and Management, and the BECC, which is the strong research area on biodiversity and ecosystem services in a changing climate at Lund University and the University of Gothenburg. Special welcome also to our online participants. So we have this hybrid seminar. We are around 20 in the room here, and we are, last time I checked, we were around 20 online as well. So great. So we will, I just say that I'm Daniel Slunge. I will facilitate this seminar. I'm leading this FRAM Center and also part of BECC. So we will start now with a joint presentation by two excellent researchers at first, we have Maj Rundlöv, who is a docent, associate professor at Lund University. Most welcome. And also Julia Osterman, who is a doctor here at the Department of Biology and Environmental Sciences at the University of Gothenburg. And then after this science presentation, we have the great pleasure here to have Andreas Gumbert with us, who is the policy officer at the Director General for the Environment of the European Commission. So warmly welcome also, Andreas. And then the last half hour we will spend for a discussion, etc. If there are some questions, we'll have like one or two questions after each presentation also. So without further ado, I think we are ready to start the presentations.
Thank you very much for inviting me to this, and I'm super happy to be here with Julia also, because although we haven't actually worked together properly, we have followed quite similar lines and interests, which is the reason why we have this joint presentation now. And before we dig into the science, we wanted to introduce ourselves a little bit. So I'm a guest here in Gothenburg now. My office is in Lund, in the ecology building on the 3rd floor where the arrow is, so that's usually where you find me. My background is in environmental science and ecology. I wrote a thesis that dealt with biodiversity in agricultural landscape, focusing a lot on organic farming and landscape heterogeneity. Then I moved on to a postdoc more on pollination, and there I also, even when I was a PhD student, I really enjoyed to work with both farmers and authorities and on questions that we as a society think are important. So this is a report on bee death that was commissioned by the Board of Agriculture. I've done postdocs both nationally at SLU Uppsala and then in the US, at University of Minnesota and University of California, and I will talk a little bit about that work during the presentation too. Now I'm also a part of BECC that Daniel mentioned, and I lead the work packages in two of my dream projects, two Horizon Europe projects. One on restoring pollinator habitats across agricultural landscapes. That's covering one sphere of my research, which is focused on conservation. The other one is Pollinera, and it deals with pesticide pollinator environmental risk assessment, and that sort of covered the other sphere, which is landscape ecotoxicology with a focus on pollinators. And I will focus more on this second sphere during the talk now. I'm handing it. It will be a bit back and forth during the talk.
Yes, because we decided to do this together. And so now I'm introducing myself. I'm Julia Osterman. I work here at BioEnv. So I did my master's at SLU in Uppsala, where I was supervised by Joachim de Miranda, and it was in a project that actually Maj designed. So that's where we first, when we never worked really together, but I worked in her project. And then I moved back to Germany, where I did my PhD more on crop pollination. So that's where the apples come from. I looked into how pollinators contribute to crop production. But actually my interest in bees started from being a beekeeper, and that's where all of it started. And then I did 1 year postdoc at the University of Freiburg, where I coordinated the project application for RESTPol, which is where my is now leading work package 2. So again, somewhere connected but not really working together. And now I'm at the University of Gothenburg, first as a postdoc and now as an independent researcher. And now I moved more towards wild bees and wild bee conservation.
You all know that pollinators, they are mainly insects, I assume, or? Nod, no, shake your head. Yeah. Most of them are insects, but they're also from other vertebrate groups. But in Europe, pollinators are mainly bees, butterfly, hoverflies, a bit of carabids and wasps, and then under butterflies you can also count the moths. But looking at the contribution of pollination services to ecosystems and to crops, it is mainly focused on bees, even if non-bee pollinators also contribute. So if we want to quantify that from a human perspective, 75% of the crops that we grow, they are in some part pollinated by animals. And also about 1 to 2% of the global GDP is provided by these pollinated crops. But they also, the pollinators are key functions of ecosystem when they pollinate wild plants. Most of the flowering plants, they depend on insect pollination to set seed or produce fruits. But they also, of course, as all biodiversity, they have an intrinsic value, so they also are valuable in itself, and that's what is brought up in the 1992 Convention on Biological Diversity. And most excitingly, there's also a nature restoration regulation that was approved last year in August, and I think that that's giving us an opportunity to work with biodiversity, and both important for itself and the importance for society, that an opportunity that we haven't had for 30 years. And most people have heard about the pollinator crisis, and I think that today pollinators have the same sort of societal view on it as birds had in the 70s. So there was EU directives on birds in the 70s, and now we have a nature restoration regulation that has an article focused on pollinators. It says something, the way we make these regulations and law, it says something about what society values currently. And here are bees, but butterflies are the only pollinator groups that we have a systematic monitoring of. One of the indicators of the status of nature comes from these structured annual monitoring across EU member states of grassland butterflies. So just like to have an index for how economy is going, you have an index for how nature is going, and it's not going very well. Over the period from 91 to today, where this grassland butterfly indicator has been calculated, there's been a 50% decline. Sweden started to contribute with grassland butterfly data in 2010, where this butterfly monitoring started in Sweden. And as I mentioned, pollinators have their own article in the nature restoration regulation. It deals with restoring pollinator populations, but it's also a part that deals with pollinator monitoring. So eventually we'll have the same type of indexing for pollinators as we have for grassland butterfly to see how well we fulfill the goals of the article. But now we're not here to talk about only pollinators, it's pesticides and pollinators together. And the reason for that is that one of the driving forces for the decline of pollinator is pesticides. In this global assessment, experts have judged that it's an important driver across the whole globe. And there are several meta-analyses that have quantified this threat also. So this is one of the more recent ones published this year, where you have collected a lot of studies to figure out what is the importance of different drivers. You can see them, can't see them. You can see them to the left in the slide, that pollution is where pesticides fall, and that is in relation to the other one quantified as the most negative driver for local biodiversity. And it's across both biomes and organism groups. The pollinators that fall in the invertebrate category here, and you can see that that's separated from the zero line. So here it is a decline of local biodiversity of invertebrates, mainly on a local scale. When talking about pollinators and pesticides, it's good to understand that what ultimately leads to an impact actually comes from how we regulate pesticides, so what is allowed to be used, and that's where the risk assessment comes in, that we will talk about a little later. But it also depends on what farmers choose to use, which relates to the pest pressures, the sort of values for society, and how farmers also perceive both pests and pesticides as a tool for pest regulation. And information is really a valuable part that we'll come back to too. But that is how pesticides end up in the environment. But then you have the pollinator activities too. And it's when these meet, the pesticides in the environment and the pollinators activity, that's where you get the exposure. And that's the prerequisite for any effects. An effect is when you bring together exposure and how toxic the different pesticides are, and then you can have effects on pollinators and eventually on the pollination services that they provide. I want to zoom in on the latter part, because we've been part of trying to make a roadmap on how to improve environmental risk assessment for chemicals and pollinators. An essential part of that is to understand how this intersection of the pesticides in the environment meet the pollinator activity. And there we call it external exposure. And then that leads to how individuals of pollinators are sensitive. And Julia will come back to that a little, or I will come back to that. We'll come back to all the parts. How individuals are affected, how they can detoxify, how they can handle different stressors and pesticides in particular. But then if you take together all the individuals, you also have the population resilience. So all the individuals together, because you don't lose a species by losing a few individuals, and population can recover or be resilient to these stressors, especially if they have the conditions for it, the landscapes and the beneficial aspects. And what we know around these different aspects, exposure, individual sensitivity, and population resilience, is very bee heavy. And we will also talk about bees mostly today, because that's where we have the knowledge. And the knowledge is also more focused on this intrinsic sensitivity, how individuals are sensitive to pesticides. And that's also one of the core parts of the environmental risk assessment. We know less about exposure patterns in reality, and we know much less across the whole different aspects on butterflies and moths and hoverflies. It can also be good because these colors, the redder, the more activities we spend, publication, projects, all of those things we spend on an intersection of topic and an organism group. But they are not scaled to how many species we have of these different groups. So in Sweden and in Europe, we have one honeybee species, almost 300 wild bee species and hoverflies, and several thousands of butterfly and moth species. So our activities are very much biased to very few species. And you can see that also if you look at the scientific literature and how that spreads across bee species. So the green part is focused on the honeybee, Apis mellifera. It's just one species. And then the second most explored group are other managed bees, and I will talk about them. They include bumblebees and solitary bees of few species. Also good to keep in the back of your head. And this number is for the European Union, the total number of bee species, almost 2,000. So bees, even these managed bee species, represented by three specific species, they are now part of a revised guidance on how to do risk assessment of pesticides within the European Union. So before, or actually still, we only have the honeybee as a model for all bee species, all the ones in the European Union. But in the revision of this guidance, that it's approved by the European Food Safety Authority that developed it, but it hasn't been implemented in the European Union yet. It's still work ongoing, and that's why it's so timely to have a seminar like this, and also have Andreas invited from DG Environment. But these three managed, or we can manage, they are also wild. Bombus terrestris and Osmia bicornis, a bumblebee and a solitary bee. They also occur in the wild here and in many parts of Europe. But we can also manage them, so we can buy a bumblebee colony or cocoons of the Osmia species, then put them out to test in the lab. And that's the convenience of the honeybee also. It's beekeepers keep it, so it's easier to do experiments, and you will talk about that also. But even with these three species, we have a lot of different traits, so they have different lifestyles, and we will come back to different aspects of these lifestyles, which include different sociality, how big colonies they form, or if they form colonies at all, if they communicate around where resources are, how far they go in a landscape for foraging, and how many plant species they include in their diet. When neonicotinoids was on the policy agenda, when the European Union were trying to figure out, should we allow the more toxic neonics, or are they too much of a threat to bees in the wild? There was a lack of understanding about how neonicotinoids of this type, in this case it's clothianidin, which is one of the more toxic ones that we don't use anymore in the European Union, but it's used globally still. And there was a mismatch between what you saw in the lab and what you saw out in reality. So it's not surprising that an insecticide kill an insect, a bee, because I mean they are designed to kill them. The question was rather, what are the consequences of using it the way it's regulated, in the concentrations it's approved, in the crops that it's allowed for use in, for the pests that it's going to target, so the actual uses. And we got a commissioned work from the Board of Agriculture to put pesticides to the test in the most risky way. And it was this seed coating with clothianidin in full flowering oilseed rape fields that was sown in spring, because then it's a short period between when you seed the coated seed and then it leaks out in the soil and then it's taken up by the plant and it protects the plant from pests. And we did that, and we collaborated with authorities and farmers that did this on whole fields or seeded untreated seeds. What we saw was that for honeybees, we could not see a difference in colony strength, which is one of the parameters that you use in environmental risk assessment, focusing on honeybees. But for bumblebees, the two species, and the solitary bees, the two representatives of non-apis bees, wild bees, they had reduced production of queens and males, which are the ones that it's a reflection of reproductive success, and it also says something potentially about population level effects. For the solitary bees, they are alone, so they need to produce offspring, otherwise they will not have any offspring next year. And here, the difference was that there are many differences, but here the honeybee, they could tolerate this level of contamination of the landscape, while the other species and wild bees in general, they could not. So in fields that were treated with the neonic, we saw half the density of wild bees compared to in the control fields. So the honeybee, they can tolerate it, and they also have this social buffering, because they have colonies that are tens of thousands of workers with a queen that is protected. They can lose a few workers and still keep up with the production of new workers. They also don't have to reproduce every year. So social buffering and tolerance are probably contributing factors here. But this was on a single neonicotinoid, a single insecticides. And bees in agricultural landscape, they are for sure exposed to a mixture of pesticides, and actually to a mixture of pressures and enabling factors. But for us, it's been really important to try to see if we can quantify this mixture pesticide pressure at a relevant scale for the bees. Because bees, they forage the landscape. They are central place foragers. So they have their colonies or nests at the central place most of the time, and then they forage the landscape. Different size depending on if they are, the solitary usually have smaller foraging range, and the bigger the colonies, the bigger your foraging range are, up to several kilometers. And in this study, we had the centrally placed nests and colonies, and then we allowed the bees to sample the landscape, and we sampled the colonies for pollen. And pollen is one of the two main food sources, along with nectar. Pollen is useful because we can easily collect it without killing the bees, and then we use it to quantify pesticide residues in the pollen. So in the food, it's the same as we do for human foods. There is a quantification in different food items, and the principles are similar here with pollen. But different substances, different pesticides are different toxic. So if you would just pull together these concentrations, you would weigh something that is very toxic similar to something that is very untoxic. So when we talk about this landscape risk, we calculate it as an additive toxicity weighted concentration. And we do that by quantifying pesticide residue in pollen for different pesticides that come from different fields, but they are integrated by the bee foraging. Then we add information on toxicity, which usually comes from honeybees because they are part of the risk assessment, so it's mandatory to produce this toxicity information for the honeybee, but not for bees in general. Different pesticides have different toxicity, so we divide the concentration by the toxicity to create toxic units, because then you have them on an equal, you can compare them. And then we add them up, what we find in a sample. So then you have a representation of the different pesticides, how much there is, how toxic they are, and how they sum together. And even though the 3 bee species have quite different foraging ranges, from a couple of 100 meters to a couple of kilometers, their calculated risk based on their pollen and honeybee toxicity is quite well correlated for this type of ecological data. So even if you only would sample pollen from honeybees, Apis mellifera, it also says something about the risk to the other 2 bee species, which is useful because it's so much easier to collect pollen from honeybees compared to the other 2 bee species. And another thing is that, a risk indicator like the landscape risk based on pollen, it's only meaningful if it indicates what you intend it to indicate. So we're trying to put this risk indicator to the test to see how well it says something about how bumblebee colonies grow, how they produce new queens, how the solitary bees create offspring. And it seems to be quite a good predictor. There's lots of variation, but it seems to predict with increasing risk, you have a decreasing reproductive success and colony growth. So here it seems like it's meaningful across a few bee species, at least to calculate risk this way. It is also useful because it integrates these different pesticides that are part of the environmental contamination of relevance to bees. If we take the information from this cross-European study that had more than 100 sites where we had bumblebee colonies across 8 European countries, and where we collected pollen to estimate the risk. If you look at that pollen in relation to local crop production, which was either apple or oilseed rape, we can see that across all the samples, 95% of the samples had more than 1 pesticide in the pollen sample. It was on average 8 substances, ranged between 1 and 27. If you, across the whole material, looked at what you found at apple orchards, there were 80 unique compounds, and at oilseed rape fields, 68. And the risk was on average also higher if you were at an apple orchard. But it's very well supported that pesticide risk do not come from a single field. It actually comes from many fields, from the landscape, from the foraging landscape. You can't predict the pesticide risk by only looking at single fields. Having this type of risk indicators that integrate the different mixtures across the landscape and that are biologically meaningful, so it predicts a biological effect on the organism you want to protect, is also useful for fulfilling goals that relate to national action plans on sustainable pesticide use, because they are all the EU member states, they have to have these type of national action plans, and they sometimes have goals that are specifically related to pollinating insects. And here, to track the goal fulfillment, they are usually the sales of pesticides or the education of farmers on how to use pesticides well, but there's no indicator for pesticide risk that is specific for pollinators, and this would be one that you potentially could use. But it's only indicative for a few species of bees, and we don't know how general it predicts pesticide risk to pollinators. Fortunately, that is one of the goals of the Pollinators project, where we have, in one of the specific objectives, we want to develop and test the pollinator pesticide co-monitoring scheme. So you monitor both the pollinators and the pesticide drivers together. But that's ongoing, so I can't say about anything more specific about it. What I can say, and I want to highlight, is when we're doing environmental risk assessment related to pesticides or any environmental risk assessment, specific protection goals are very important, because they are sort of thresholds that we use to assess if any use is predicted to exceed an acceptable risk, or if it falls below, and then it can be approved. And to come to these specific protection goals, you sort of want to know, or you need to know, what is the normal, how, in this case, it's bumblebees, and it relies on data from the same cross-European project. Here we wanted to play with the data to see if we would develop this type of specific protection goal that doesn't exist for bumblebees yet. It only exists for honeybees, and for honeybees the specific protection goal that is suggested in the revised bee guidance document is that it shouldn't be more than a 10% reduction in colony strength in honeybees. If it exceeds that, then it can't be approved. But for bumblebees and solitary bees, the specific protection goal is actually undefined, because we haven't been able to determine an SPG. Using this data, we took the 25th lowest risk quantile information, calculated what that would mean in terms of the colony weight, which is a really good predictor of bumblebee colony performance. And then that baseline 10% reduction in colony weight was what we played with as a specific protection goal. And if that would be the specific protection goal, 60% of the colonies in this study would actually fail in the risk assessment. Or they will be too risky, under too risky conditions. Because it covers different European countries, it also says something about the risk situation for pesticides in the European Union, which actually has a really good environmental risk assessment system that at least try to do something beyond the honeybee. There's also lots of ongoing work within the European Food Safety Authority. As I said, they led the development of the new and revised bee guidance, but they are also working on new guidance for other terrestrial organisms, where they try to deal not only with the direct effects of pesticide on organisms, but also take in the indirect effects. And that is when an organism interacts with another organism that is directly affected. But that is also ongoing work. And finally, I wanted to mention a bit more positive things, because pesticides will be part of agriculture production for the foreseeable future. There was, similar to the nature restoration regulation, there was a regulation suggested on pesticide use and reducing the risk with pesticide use, but that was not supported, so that is not something that is taken further in the legal framework of the European Union. So we probably have to work more with mitigating pesticide exposure and effects, and one way of doing that with bees seem to be to provide flowers. And Julia will come back to dietary aspects too. Here it's work from California, so part of one of the postdocs that I did, where we looked at one solitary bee and a bumblebee species, where for both species local flowers increased their reproductive success, but in the bumblebee it also mitigated effects of pesticide risk. So if there were no flowers, if there were any pesticide risk, there were in essence no reproduction at all. If there were some flowers, you actually removed this pesticide risk part, so there was at least some reproduction. Not a huge amount, but still it brought it up to the same level as if you wouldn't have any pesticide risk. And if you expand that to think about how agricultural landscape look, we draw from a cage experiment that was done by a PhD student that were in the same lab as I visited in California, where they have pesticide and flower resources crossed in a cage experiment. And if you apply that to agricultural landscapes, in the top picture where there's abundant flower resources across the season, but very little pesticide exposure, here you would predict a population growth rate of 2. But then as you move down and have lots of flowers, but high pesticide exposure, you have a population growth rate of 0.5. So the bees, they don't replace themselves even if you have lots of flowers. But it's even worse to have no local flowers, independent on how much pesticide you have. Because if you don't have the flowers, the food, it doesn't really matter if you have high or low pesticides. You don't have the conditions for almost any population growth, estimated to 0.2 here. So it's better to have flowers and pesticides than it is to have no flowers independent of pesticides. But it's best to have no flowers and lots of flowers and no pesticides, because then the population can replace itself. But that was in California, and this is Sweden. And this is Sweden now, where farmers this year have been able to apply for financial support to put in flower strips or flowering fields with at least 3 flowering plant species, and they get quite a lot of payment for it. So it's become quite attractive. And in the southernmost part of Sweden, in the agricultural plains where there are almost no flowers, about 3% of the agricultural land have been planted with these types of flowers, or the cropland. So this shows that if you pay for it, you will get flowers in the landscape. But the question is, what is the benefit for pollinators to have these quite few flowering plant species that provide food for, in general, for pollinators, but not so much perhaps for all pollinators? And we have a project where we want to see if it works the same in Sweden as it did in California, if the flower plantings also can be used as this mitigation for pesticide exposure. I want to end with saying that flower plantings like this can be one of the tools in integrated pest management, which is mandatory for all farmers across the European Union. And I even want to bring in the pollinators, because then they often share drivers. They are affected by the same things, the pesticides, the landscape, the flowers, both the pests, the pollinators, and the natural enemies of the pests. So you can manage them together, can create landscapes that are beneficial, can put in flowering crops and flowering strips. And then you monitor your pests and pollinator needs, and then you only use pesticides and and artificial pollination, which honeybees can be counted as whenever you have a need or you pass an economic threshold. And then, Julia, it's your turn.
I start my part with a nice illustration from one of my publications and dig again a little bit more into how foraging activity and preference determine pesticide risk. And here you have the example of the honeybee in yellow that is living all year round and is a generalist, so it forages on all kinds of things, crops, but also the flower strips and maybe even on trees. But then you have solitary bees, and some of them are very specific in their foraging preferences. Here you have an example of a bee that only goes on pumpkin, and if that crop is then sprayed, it will only be exposed to that pollen and has maybe a higher risk, while another solitary bee that completely ignores crops at all will not have a risk at all. So it's really important to think of the foraging activity and preferences, and also, as you said, range and how they communicate and so on. And then I want to dig again into our diversity that we have, and Mai has already mentioned that in Sweden we have around 300 different bee species. In Europe it's around 2,000, and globally it's estimated that we have 20,000 different bee species. So it's a lot to consider and take into consideration. In Sweden, 27% of the bee species are actually red listed, so they're not doing so well. And as we have heard of Mai, it's that bees differ in their life history traits, and we have already touched on sociality, of which the honeybee is highly eusocial and lives in the colony all year round, has a lot of workers, while most of the bees that are actually in Sweden are living solitary. But they do not only differ in their sociality, but they also have different nesting preferences. For example, some nest in snail shells. Most of the bees actually are nesting underground. One example you see here. They're different in their diet and in their activity period. So when some are just active 4 weeks in spring and some are active only in autumn. So it also depends on where they're going to forage and how they're exposed to pesticides. And I got specifically interested in the diet aspect because, as you have heard, honeybees are generalists, so they collect from all kinds of different plants. But actually many wild bees are oligolectic, and oligolectic means that they have a more narrow pollen collection, and it's defined as collecting pollen from only one plant family, but in some cases it gets even more extreme that they're only collecting from a single plant species. And here I have compiled a few examples with nice pictures from my husband. And this is Andrena vaga. It only collects pollen from salix. So that one might not be so exposed, right, if salix is not next to an agricultural field. If it is, maybe it gets exposed. Then you have here Melita hemorrhoidalis. It's only collecting pollen from campanulaceae. Here you have Chelostoma florisomne, only collecting pollen from ranunculaceae. And then this is Andrena marginata, who's only collecting from a single plant species, Zooxisa pratensis. And overall, a student of mine has collected data from all the bee species that occur in Sweden, and 24% of the bees that we have are oligolectic. So it's a quite substantial amount, and so we need to consider this diet aspects as well, because we have seen from Mai's data that diet can somehow also affect or mitigate effects from pesticides. And As you have heard earlier, I did my master project in mice project that was published in 2015 by her. And when I looked at the data some years later again, I thought it's very strange that the nesting activity was low in general when she used Osmia bicornis, which is a generalist species. And then I found another study that used mesocosms or flight cages, where they either had monoculture with oilseed rape or oilseed rape and a flower strip, and looked at different aspects of Osmia bicornis reproduction, for example, the brood cells, the number of cocoons, the development of the cocoons. And the biggest effect that that study showed was not of the neonicotinoid, which they wanted to test, but of the diet. So when they had only access to the monoculture oilseed rape, then the number of brood cells was halved compared to when they had also flower strip. So this is working with a generalistic species. And then in addition, the only effect that they found on the neonicotinoid was when they were in this monoculture, and then there was an effect on the development of the brood cells. So then I was thinking, so the Osmia bicornis is suggested to be used in the risk assessment, and then often oilseed rape is used as an example crop. But what if we're testing something on something that is not working? So maybe we need to find another model species if we want to work with solitary bees. And also maybe we should understand what effect it has on those that are specialized, so their diet is very narrow. And then in a dark corona time, I came and discussing with my husband, I came up with an idea to use this species instead, Osmia brevicornis. It might be confusing because the names are quite similar. It is specialized on brassicaceae, so it likes oilseed rape, and it actually flies when oilseed rape flowers. And it nests also in nesting material, so potentially we could use it. But you cannot just come up with using a new model species. Maybe some of you have tried this. There's a lot of challenges on the way. And of course, there's a reason for why when we want to expand from honeybees to other bees that we work with Bombus terrestris and Osmia bicornis, it's good that we start with that, because we can buy them and it's easily to work with them, while many of the wild species, you cannot get the same amount of numbers that you would need to do proper testing. So our first questions was, can we somehow rear them, and under which conditions? So we had different sizes of the tubes to see what their preference is. And also, can we work with them in the laboratory? And we managed to rear them, and we managed to feed them in the lab, and we tested their different solutions to make them eat, because if you want to expose them in the lab, they somehow have to eat. But compared to honeybees that are more easily able to feed, solitary bees often refuse to eat. They're probably more used to starvation. So we need to trigger them a little bit to eat. So we try just to put a yellow tape next to the food, but in compared to nothing at all. And then we had also a flower petal. And we managed with a flower petal that the rate of feeding increased. So we were happy that it worked, but in this test we didn't expose them yet to any pesticides. It was just plainly look, do the methods work, can we use it? And then we were lucky, and I'm very lucky that a colleague of mine, Dimitri Wintermantel, has helped me work with that species, because I got so excited, but I didn't have the facilities or the money. But then we got funding from the European Commission in Wild Posh to actually work on it. And here in Wild Posh, I'm working in this work package too. And first of all, we develop protocols together in different labs to expose pollinators to pesticides, because when you want to see how sensitive they are, you need to also use similar methods, because often the data that is out there is hard to compare. If some are feeding them, if some are using topical exposure, sometimes you don't even know all the details. So the first step is develop protocols, and now we are exposing them in laboratory condition and then in the semi-field. So Alicia, who you saw on the previous picture, she was an excellent master student who worked last year on this project where we had the two species, so Osmia brevicornis, that's the specialist, remember, on the BECC, and then Osmia bicornis, that is the generalist, and we managed to have enough numbers to do 9 treatments. But first, here you see how the workflow is. So first we have to hatch them, then we acclimatize them where they learn how to eat together in boxes of 20 individuals. Then they get exposed after a short starvation period. And here are the treatments that we exposed them to. So there was positive and negative control, and then 2 insecticides, so cypermethrin, acetamiprid, and then a fungicide, tipuconazole. And we exposed them to all the single treatments, but also the 2-way mixtures and the 3-way mixtures, because as you heard from Mai, it's very rare that they're only exposed to a single pesticide. In most cases, they're exposed to a mix. And in many cases, we don't know how they're interacting. So that's another thing we want to test. Unfortunately, I cannot show you any results of this yet, but stay tuned, it's going to come. And then as a follow-up this year, Alicia started her PhD in the same project and under the supervision of Dimitri and myself. And now we're going from the lab to the semi-field, where we are again using those 2 species and exposing them to a insecticide, acetamiprid, and a fungicide, tipuconazole, but in their mixtures. And then the combination, we had 39 flight cages. And you see here that they had different nesting boxes, which is again based on what we developed, the methodologies for what they like to nest in and so on. But since we're having a seminar in FRAM, I thought it's good to talk about the risk assessment as well, and how our research kind of feeds into it. You mentioned it many times, Mai, already. But so this is how the process is normally done. So there is toxicity tests under laboratory conditions. If there's indication of risk, then it goes to semi-field outdoor cages. And if there's also an indication of risk, then it goes to the field testing. And while in Europe, EFSA has approved the guidance to use also Bombus and Osmia species, it's not yet implemented. So most of this is still based on honeybees. And even though if we see some indication of risk on the way, if the field experiment shows it's okay, then it can get approved. So this shows 2 main problems. First of all is that if we use with honeybees, and if the field studies overrule the lab studies, then the social buffering that Mai has shown could potentially make problems that we oversee some risks that are there for the others, while for honeybees it's okay. And the second problem comes with statistics. So in the guidance from 2013, there was the protection goal of a minimum of 7% colony reduction with a power of 80%. Statistic power of 80%. And then 2 researchers in Canada actually compiled all the studies that were out on neonicotinoids from semi-field or field studies. And this is maybe a bit of a tricky graph, but the ring inside shows would show that if there would be a point there, then studies would have the power to detect a small effect, so 7% reduction, if it would be towards the center. If it's in the second ring, then studies would be able to detect a medium effect of 15% colony reduction. And if it's on this ring, then the studies could detect a large effect. So the further it goes to the middle, the better it is in detecting effects. And remember that EFSA had this protection goal of that it should not be higher than 7% reduction. But actually none of the studies could ever detect a 7% reduction, and none could even go have a medium effect. Actually, my study and then my follow-up publication, we were one of the best, or you were one of the best ones that could show an effect of maybe 20% reduction in colony strength. And it's very crazy to think that fewer than 27% of studies had the power to confidentially detect large worker losses, a critical level for colony survival. And when you have to consider this is also that an 80% power of a study means that in 20% of the cases, there could be an effect, but you can't still detect it. It's not statistically shown. So 1/5 of all the studies would not find a statistical effect. And so EFSA in the new guidelines suggest equivalence testing instead of difference testing. And MAI has been a part of this new guidance. And the benefit of equivalence testing, so instead of testing whether they're different or not, you test whether they're equal. And there's two advantages of this. First is that if you have only a small effect, but it would be significant in the difference testing, but it's not biologically meaningful, in the equivalence testing, you would get to a low risk. And the second advantage is that if you have high variation in your data, which you often have from honeybee colonies, in the difference testing, you would get a non-significant effect just because the variation is so high. But in the equivalence testing, you would get the classification as that the high risk is not excluded. So equivalence testing could be a nice add-on for the risk assessment to make it more safe for the environment. And with that, we end our presentation. So the risk assessment so far only deals with bees and only a few managed ones. So it would be good to even spread it further. Now we only also focused our presentation on bees, but as you have seen, there are many other bees out there, and we need to define specific protection goals for other organisms as well.
It's the last slide with the last outlook. The other 2 points are that bee-collected pollen is probably promising for this post-approval monitoring of pesticide risk and inferred effect, because you handle mixtures. And then also flowers or diet is an opportunity for pesticide risk mitigation. And with that, thank you very much for listening, and thank you all the other ones that contributed.
So now we have changed presentation mode here. So we have projected your slides also here on the screen here in Gothenburg, Andreas. And so now Mai said that there were, if I got it correct, like 60% of the populations were living under, bee populations were living under unacceptable risks. So the question now, is there a policy response here and what is happening within the EU on these issues. We are so glad to have you here, Andreas Gumbert, and please give us some views on this. Thank you very much.
Thanks very much for the organizers and also for the brilliant speakers before me. I learned a lot. It's really a pleasure to hear all this about all this excellent science which is ongoing on this very important topic on pesticide impact on pollinators. Daniel, you introduced me. I'm Andreas Gumbert. I work in the European Commission and DG Environment on the implementation of the pollinator initiative. And and there's a lot to say on pesticides and pollinators. So I will have three main topics. And some of them overlap a little bit with what you have heard already. So I will give an overview about where we are in the implementation of the Nature Restoration Regulation as regards pollinator monitoring. And then I will talk about some actions that we have in our EU pollinator initiative at EU level in relation to pesticides. And then thirdly, I will speak about an emerging new indicator in the global framework. Just to start with the Nature Restoration Regulation, Mai has already indicated it. This is a regulation which was adopted in 2024, and it obliges the member states to demonstrate a reversal of the decline of pollinators already by the year 2030, and to keep a positive trend beyond. And this has to go on until there are satisfactory levels reached. And how do the member states demonstrate these positive trends? There is an obligatory monitoring foreseen, which has to be undertaken on an annual basis. It has to follow a standardized protocol, and the monitoring must be representative for the situation in the member state. And how exactly the methodology is to be followed. This is laid down in the so-called Delegated Act. So this is a subordinate legislation under the regulation, which has been adopted by the European Commission a few weeks ago on the 19th of September. It is not yet enforced because the European legislators, so the European Council and the Parliament, still have the possibility to object to this draft legislation. We are very positive that they will not use this right, and which would mean that at the end of the scrutiny period, which will be on the 19th of November, in a few days, so the Commission will be able to publish this delegated regulation in the official journal of the EU legislation of the European Union, which will make it applicable for all the member states. So what do the member states have to do? They will have to monitor the four main groups of pollinators, which are wild bees. So we only focus on the wild bees, hoverflies, butterflies, and large species of moths. This monitoring will lead to the calculation of different type of indicators, on the species abundance indicator, and on an indicator on the diversity of pollinators at each site. And this can only be calculated for relatively common species, but there is also a monitoring module focusing on highly endangered species. So, and this will feed into a total species richness indicator, counting all species that are seen in a country in a given year. So all specimens which will be recorded will have to be identified to the species level. And the monitoring has to take place on a predefined number of monitoring sites. There is a minimum number in each country, which the smallest number is 30 for Malta, and the largest number is 120 for France. And these sites are to be allocated in a randomized manner, but there is also a stratification exercise so that the ecosystems are represented in a proportion as they occur in the member states. So then there will be transects walked and light traps for the night active species. So with this monitoring, we hope that we'll get long-term trends, which will, on the one side, inform us, of course, about the well-being of the pollinators, but on the other side, that there will also be a wealth of scientifically relevant information that allow, for instance, the co-monitoring with the key drivers, such as pollinator such as pesticide use, but also other drivers. I'm now coming to the second part of my presentation, and that concerns the activities that we have in the EU Pollinator Initiative. So this initiative is a policy. It is not a binding policy, so we can consider it a soft policy in the sense because it is a strategy paper the Commission has put forward, which is only binding the Commission itself. So it includes an action plan of more than 40 actions, and 6 of these actions relate to pesticides. Mai has already indicated the importance of integrated pest management. So the Commission is supporting, in particular, Horizon Europe projects, research projects that will expand the toolbox available for alternatives to chemical pesticides. And in particular, we have now supported under the LIFE instrument a project which is called AgroWise, which will help the member state to bridge the gap between, on the one side, integrated pest management, which is a principle laid down in the Sustainable Use of Pesticide Directive, but it's not easily applicable for the individual farmers when they have to take concrete decisions on what exactly they are supposed to do. And AgroWise will hopefully help the member states to define the rules for this. There was already information provided on the importance of indicators. We speak about that in the third part of my presentation, so maybe I skip it for now. The next action concerns so-called emergency authorization. So what are these? These are the member states have the possibility to use or to approve at a national level the use of plant protection products that are not necessarily authorized at EU level, or that are also authorized only for a certain use, and so the member states can expand beyond these existing authorizations. So they have to notify these emergency authorizations, which should be exceptional, but unfortunately we have those every year, to the Commission. The Commission is monitoring whether there's a justification for those, and it has now started, for instance, one case, an infringement procedure, a legal procedure against Romania for the use of neonicotinoids in combination with sugar beet production, which is going against EU legislation, according to the views of the Commission. The next action concerns the implementation of the FSA-B guidance document. Also there, it has been mentioned already, so we have this new FSA-B guidance document, which is relevant for expanding or improving the environmental risk assessment of pesticide risks for bees. And the Commission is currently preparing the implementation of this in a set of legal acts. These are the Commission regulations, which are mentioned here, which have to be amended. So a few weeks ago, the publication of the draft changes was launched, or actually the possibility for the public to make comments expired in early October. And what is good here is certainly that the risk assessment will be expanded to wild bees. So at the moment, the honeybee has been the key indicator species among the pollinators. So, but we are suffering, by the effect of an undefined specific protection goal. This is something that has been said earlier. So while it is precisely defined what is the maximum level of mortality, the maximum level of damage that may occur for honeybees, this is not the case. We don't have such a quantitative objective defined for the wild bees, and that leads to a situation where the risk assessment cannot be undertaken in the same way. For instance, there is not a possibility for a tiered risk assessment where you would start with laboratory tests and then you would move on to field tests if this is necessary. And as a result, the tests with wild bees are only necessary in case that the existing established monitoring species, which are honeybees or other non-bee, non-target arthropods, already indicate the risk. And that is a concern, and we can only overcome that in the long run with the definition of the specific protection goals for wild pollinators as well. I will skip the other 2 points because I think they are maybe a bit too technical for today's discussion. But I would like to add another a bit technical element, but I think it is an exciting and interesting new development, and that is the development of a new indicator for pesticide risks, which could be useful also for informing us about threats to pollinators. And what you see here are existing statistics and indicators that we are having for pesticides already at the moment. On the left side, you see the sales of pesticides, all pesticides lumped together, so total kilogram of used active substance. And you see that basically there is not much developing. The sales are pretty constant. There has been a recent decline since 2021. If you look at all pesticides, and you see quite a substantial increase in the use of insecticides if compared to 2011. This is not telling us a lot because we don't know in these statistics the actual toxicity of the pesticides being used. There is a harmonized risk indicator. It is an indicator that is shown on the right side. It is an indicator that quantifies the total risk emerging from the use of pesticides, but also this there is, first of all, there are some question marks around the methodology applied for this indicator, but then also it is not very differentiated regarding the impact on different type of organisms, and therefore there is an interest in developing something which is more fine-tuned with respect to the biodiversity impact. Under the UN framework, under the Convention on Biological Diversity, there has been in 2022 a large agreement reached, the Global Biodiversity Framework, which is more or less the Paris Agreement for what's the Paris Agreement under climate legislation, under the climate change convention, is the GBF is under the Convention on Biological Diversity, and this has included a target to reduce by half the risks from pesticides at a global level, and this will be informed by a so-called headline indicator. And the methodology for this headline indicator is currently being developed by the FAO. And there is one candidate of 2 possible indicators, is the so-called aggregated total applied toxicity indicator, or ATAT. I'm presenting here some work that the Commission is undertaking, that the scientific branch of the Commission, the Joint Research Center, is undertaking. Here, we don't have to go into the mathematics and all the details, but what this indicator basically does is it sums up the combined toxicity of all active substances for different groups of organisms, and that allows us to basically quantify much better the projected biodiversity impact on these different group of organisms. If we look at that for one example, one member state here, then you see basically that since 2011 there has been a slight decline in the total applied toxicity, which is not weighted. So basically we're just adding up here all the toxicity effects across all different group of organisms. But these changes are they are not uniform. So we see first of all, we see quite year-on-year fluctuations, and we see remarkable effects for the different group of organisms, and the risk can also go up for some of these organisms. On the right side, you see here the effect on the pollinators, and we would conclude that since the phase out of the neonicotinoids, or at least some of the neonicotinoids after 2018, the total applied toxicity has substantially decreased, which is of course a very good sign. What this methodology also allows us to do, and that is actually my last slide, is that it will allow quantifying the total effects, it will allow identifying the key substances that are relevant for that are the relevant driver for different groups of organisms, and this is highly informative information to drive policies, to focus in particular on mitigation measures in order to not to address basically pesticides as a whole, but to zoom in on those areas, those crops on which the pesticides are applied, and the particular type of pesticides that are being applied, that would allow us to address the issue of pesticide risks much more fine-tuned. I stop here, and I'm very happy to answer any questions. Thank you.
Thank you very much, Andreas, for a very interesting presentation. I think this seminar has very much illustrated both what BECC is about and what FRAM is about, and I particularly like this combination of drivers and toxic units and like a more complicated risk management perspective than looking at one substance at a time and one species, etc. And we also touched upon the management strategies here, which could perhaps be a topic of a future seminar. So thank you very much for your great presentations. And before we close, I would just like to inform that we have another upcoming seminar here the 4th of December, where it's actually part of what we call the Healthy Planet initiative here at the Faculty of Natural Science and Technology, where we will focus on the triple planetary crisis and how we can safeguard science in multilateral negotiations. What can we learn from IPBES and from IPCC for the new panel that has been formed to the Intergovernmental Science Panel that has recently been formed to tackle issues related to pollution and waste. And we'll also touch upon the plastics negotiations. So very welcome to that. 4th of December, you can look at our homepages and see information on that. But thank you very much for this seminar, for joining this seminar today, and I hope to see you soon again. Thank you.